Hello dear student, myself Dr. Prashant Kumar Desai. Today we are going to discuss about the transfer section of the Ceylon cinnamon bark. Ceylon cinnamon bark is also known by the one another name, that is the Sri Lankan cinnamon. There are the two names of the drug, Ceylon cinnamon and the Sri Lankan cinnamon. The names are different but they are the same genus and the same species plant. One more species of the cinnamon that is known by the common name, that is the Chinese cinnamon. Ceylon cinnamon and the Chinese cinnamons are both can be differentiated on the basis of their morphological characters as well as the microscopical character. So here in this slide we are going to discuss about the transfer section of Ceylon cinnamon. When we look at the whole structure, cellular structure under the microscope, the whole transfer section of the Ceylon cinnamon is divided in two segments. First segment, that is a pericycle layer, or it is also termed as a stone cells, and the another, that is a secondary phloem. The complete transfer section of the cinnamon bark divided in two segments, pericycle layer or secondary phloem. The stone cell layer, the meaning of the stone cells are those cells which are found in the compressed form, and the type of the cells are found in a sclerenchymatous cells. In another word, we can say the stone cells are the compressed form of sclerenchymatous cells. First, we discuss about the pericycle layer. Pericycle layer mainly contain two type of the cellular component. One is the pericycle fiber and the another is a sclerids. These are the two cellular components. The pericycle fiber are found in number that is around 6 to 15 number are present, and they are lignified. The meaning of the lignified cells, that they contain the lignin. They are also termed as the dead cells or the sclerenchymatous cells. And the one another part of the pericycle layer is a sclerids. Sclerids are also the lignified cells, U-shaped, and the isodiametric type of the cells which contain the starch grain. When we talk about the pericyclic fiber, these are the bunch of grape-like structure. Here in this structure you can see these are the bunch shaped structure. And when we talk about the sclerids are the U-shaped type of the structure which are isodiametric in nature. So in the pericycle layer, mainly two type of the cellular structure are present, pericycle fiber and the sclerids. Now we come to the second part of the transfer section of cinnamon bark, that is the secondary phloem. Secondary phloem also contain number of the cellular component. We will discuss one by one. First, we talk about the most of the secondary phloem structure is made up of parenchymatous cells. The yellow color cells, these are the parenchymatous cells which contain abundant starch grain. Parenchymatous cells are those cells in which the intercellular space are present, isodiametric in nature. They are the living cells and contain the reserve food materials. Other cellular structure which is found in a characteristic feature, there is a presence of the oil cells, and the oil cells, they contain the volatile oil. These are the big and isolated. These are the characteristic feature, that is the big shape of the volatile oil containing glands are present here. And another characteristic feature which is present in the secondary phloem of the Ceylon cinnamon, there is a presence of the mucilaginous cells. These are the pink color mucilaginous cells are present, which can be identified with the help of the one of the staining reagent that is known by the name ruthenium red. Other characteristic feature, cellular character, that is the presence of medullary rays. That is the presence of medullary rays. This structure is the medullary rays. Some information regarding the medullary rays. These are the biseriate in nature. The meaning of biseriate is that biseriate is made up of two words. Bi matlab two and seriate means multicellular. When the number of cells are arranged in two layers, this type of the arrangement of the cells is termed as a biseriate, and they also contain the starch grain. In the secondary phloem segment, there is a presence of the some more feature that is known by the name phloem fiber. These green colored cells are termed as a phloem fiber, which are also isolated and the lignified. So secondary phloem contain number of type of the cellular structures, the presence of the medullary rays, presence of big and isolated volatile containing gland, mucilaginous, abundant of the starch grains, and the phloem fibers are also present. So this is some information regarding the Ceylon cinnamon transfer section. Some important note regarding the transfer section of the Ceylon cinnamon transfer section, that there is a absence of cork and cortex. It is the most important point. Number of times the question asked in the competitive exam, the which part of the cinnamon transfer section of cinnamon bark has been found absent. The two part names, the cork and the cortex, has been found absent. And the presence of calcium oxide crystals, Ceylon cinnamon calcium oxide crystals are present in the form of acicular raphides. Acicular means needle shape, and rapide is also meaning of the needle shape, but they are been found in a cluster form. So this is the important information that is related with the transfer section of the Ceylon cinnamon. We can summarize the whole transfer section once again. The whole transfer section of cinnamon bark is made up of two component, pericycle layer and the secondary phloem. Pericycle layer, it is made up of the stone cells. Stone cells are the compressed form of the scleral cameter cells. Pericycle fiber are present in the form of the bunch of the grapes. They are found in number 6 to 15, and they are the lignified. They are found not in a permanent form. They have been found in a scattered form. And the one another structure is sclerates, which are the lignified cells. U-shaped structure contain starch grain and the isodiametric. They are part of the pericycle layer. And the secondary phloem of the Ceylon cinnamon transfer section contain number of the cellular structure, just like parenchyma cells, medullary rays, abundant starch grain, phloem fiber, big and isolated volatile containing glands. And the important part is that there is the absence of cork and cortex has been found in the case of the Ceylon cinnamon or the Sri Lankan cinnamon. And the types of calcium oxide crystals are present. They are the acicular raphides. Thank you.